virtual Catalyst. Heather Dewey Hagborg, who did a very early Catalyst program with us in 2013 with the Broad Institute's Bang Wang called Genomics, is with us this morning. We are asking our participants what they're up to, how their respective conversations have stayed with them, if they have, and what they are dealing with in the current moment. This morning, I'm having that conversation with artist Heather Dewey Hagborg. In contrast to what we normally do, the conversation is between the two of us. Heather is an artist and biohacker who's interested in art as research and technological critique. Her controversial biopolitical art practice includes the project Stranger Visions, in which she created portrait sculptures from analysis of genetic material like hair, cigarette butts, chewed up gum, collected in public places. Heather has shown her work at events and venues around the world, and her work is held in many public collections. She holds a PhD in electronic arts from Rensselaer Polytech Institute and is a visiting assistant professor of interactive media at NYU Abu Dhabi, an artist fellow at AI Now, an artist in resident at the Exploratorium, and is an affiliate of Data and Society. She is also co-founder and co-curator of Refresh, an inclusive and politically engaged collaborative platform in the intersection of art, science, and technology. She says that since 2001, quote, I've been working at the intersection of art and science with an emphasis on concepts of the natural and the artificial. Drawing from the diverse field, including biology, computation, sculpture, and critical design, I view art as an open field in which any discipline can become subject and material. I utilize art as practice-based research with which to probe the deep and often hidden structures of media, technology, and science that dominate the contemporary moment and frame our cultural imagination. Heather is now going to share her work with us, and we'll have a conversation following that. And as Eduardo said, you can join in through chat or by raising your hand. So with that, I give the screen floor to Heather.
Thank you. All right, I'll just share some slides. Look good?
Perfect.
Okay, wonderful. So hi, I'm Heather Dewey Hagborg. I'm an artist and a biohacker, and thank you for having me back with Catalyst. So I've worked a lot with DNA in my artistic career. I've made work algorithmically generating 3D portraits, 3D printed portraits of strangers based on DNA that they leave in public. So things like cigarette butts and chewed up gum and hairs. So this was a body of work called Stranger Visions, which I talked about in the first Catalyst visit. And I've worked with the whistleblower, Chelsea Manning, to show how many different interpretations of the same DNA data are possible. So here you see 30 different interpretations of Chelsea Manning's DNA made from exactly the same data, just looking at different possibilities, different probabilities, and giving space to some of those more minor interpretations alongside the more dominant ones. But I want to focus today on recent works that explore biotechnological futures and intimacy from a solo show that I had last year called At the Temperature of My Body. This exhibition is a meditation on intimacy, desire, and spirituality, anticipating a world of emerging biotechnological rituals. The exhibition consists of 3 works corresponding to 3 different phases of love: desire, attachment, and grief: T3511, lovesick, and spirit molecule. Through the frame of speculative narrative, they introduce science that's entirely real, that's entirely of the present. Each of the works is strongly narrative and also deeply personal, and they also each raise significant ethical and political issues. By sharing a clip, an introduction from the work T3511.
A cell is a history. A cell is a home. A cell is a whole. A cell is a cage. A cell is an electric fence, enclosing your legible unknown. To break the cell, is to trespass, the most intimate of spaces. Dear donor T2305, I received 5 milliliters of your saliva in June. It wasn't hard to get. Using my academic email and mailing address, I simply said it was for research, genetic testing, the truth. When it arrived, I moved it quickly to the freezer to keep it fresh and alive. I sent 2 milliliters of your fluid for sequencing, and then I waited and waited and waited. 6 weeks came and went, and then another time over. The company was experiencing unusually long processing times. When the results finally trickled in 6 months later, I devoured your profile in a single sitting. You are 46 years old with dark brown eyes, a full head of brown hair, and few freckles. You toss and turn in bed, and you enjoy a savory midnight snack. You like to run, for miles, for hours, and this keeps you slim, keeps your heart beating strong, and helps tame your sometimes wild thoughts. You were my first, and I think of you frequently. I imagine your face, your voice, the way you walk. I'm curious where you grew up. I worry for your health, and I wonder what you would think of me poring over your genetic details. I'd like to think you would be flattered, but you might be very angry.
So that's just a little teaser of the full video, which I'm happy to share if anyone is interested in watching the full thing. So what you just saw is an excerpt of the film T3511. It's a 4-channel video installation, so you can see here one image that gives you a sense of what this might look like in an installation space with 4 screens, at different times showing the same thing, at different times showing different things on the different screens. It's a science fiction detective story romance about a biohacker who purchases an anonymous donor's saliva online, profiles their genetic data, and proceeds to fall in love with them and maybe become obsessed with them, you would say. And it's sort of a true story. So it's a semi-documentary fiction kind of blend. And what I would just mention about the installation as well, while we're on this slide, is that what I wanted to convey in the 4-channel installation was the feeling of being inside the biobank, because the work is also a meditation. We're all part of these different biobanks. We've all given some kind of sample of blood or had a biopsy or something like this, where we end up in a freezer somewhere, and that those cells, those fluids, those data have a kind of uncontrollable future. And so the T3511 is very much about these futures of fluids and cells and data. And so here you see the installation sitting literally inside of this giant robotic biobank. We can talk more about that, but this was filmed at the National Biobank in Denmark. So as for the true story part, so on June 20th, I bought a saliva sample from one of a slew of companies that sell specimens from single human donors online. Here you can see some of these examples of different kinds of things that you can buy. I chose saliva because I knew it would be easy to hack. So with very little effort, with no lab, I could just pour the stranger's fluid into a standard kit purchased in a drugstore for direct-to-consumer genomic analysis. I'm sure many of you in the audience are familiar with 20. It's the most prominent of a spectrum of companies that have begun offering genetic testing directly to consumers outside of a medical context. So you buy a kit like this for about $100, spit, drop it in the mail, and a month or so later, you get some elaborate report detailing your ancestry, maybe detailing genetic traits, health risks, things of this nature. And then you can take that data and put it into other sources to get other kinds of analyses and interpretations. So I took the stranger's saliva, I poured it in the kit, I mailed it in. I didn't really expect it to work, but it did. And so this is their 23andMe profile. So you can see they have dark eyes, they prefer salty snacks, they don't have much back hair. And when I got their data back, I began poring over it and imagining this person, what they might look like, what they might act like, how they might feel about me analyzing their genetic material. And I wanted to see how it would be, not only to imagine a person based on their genome, like I did in Stranger Visions, creating this portrait, thinking about what they might look like, but to actually make a guess at who they are. So this was around the time where there was research coming out from the scientist Jan-Yves Ehrlich showing that it was pretty easy to make an inference about someone's last name, for example, from looking at their Y chromosome. And so I basically repeated these experiments. So Y chromosome analysis led me to a close relative of the donor, a last name, a geographic location, through a very public Facebook profile. And it turned out, perhaps not surprisingly, that the donor lived, at least who I think what the donor was, lived in the same city as the company that I bought the saliva from, just miles from their lab. So by simply buying saliva that's supposedly de-identified and using easily available commercial services, I was able to re-identify this individual within a few months. So this is a kind of multi-layer story. On the one hand, it's a story about genetic vulnerability, that your data, that your cells, that your body is up for sale, that your bio data is like your other various electronic data. We've all become, I think, familiar with having conversations around data in relation to electronic companies like Google, like Facebook, but we rarely draw the connection to the parallel of our bio data, which is more kind of hidden in these, in the medical industry, for example. We don't think about it as being connected, but it is just the same data, and it is in many cases much more personal and quite vulnerable. And so your biodata, like your electronic data, is hackable, legible, and draws a very vivid portrait of who you are. But T3511 is also a deeply personal story on two levels. So first, it describes this actual experiment that I conducted to see how easy it would be to re-identify a person from saliva, from DNA. And the narrative details my own conflicted feelings about this work and kind of the questioning of what the ethical frame is around doing this kind of what you might see as white hat hacking work, kind of performing something which shows these vulnerabilities or risks within a system. And then on the other hand, it is just about love. It is a story about how it feels to fall in love, to open yourself, to make yourself vulnerable to another person, and how every love story is a kind of detective story where you're piecing together clues about who a person is. And as you learn, when you first meet them, they're this stranger, and you start to accumulate these little bits and pieces that tell you who they are. But mostly you're projecting your own wishes and desires onto them. And so T3511 is very much mixing these together. So the next project I want to share with you is called Lovesick. This is the outcome of an art residency that I completed in the spring of 2019 at the Science Center, which is a biotechnology incubator and Integral Molecular, a vaccine and drug discovery company based in Philadelphia. So together with the scientists at Integral Molecular, we invented a custom retrovirus, which infects human cells and inserts an extra copy of the gene that produces oxytocin. The hormone oxytocin is implicated in feelings of love and bonding, the promotion of empathy and connection. It's this hormone that's released, for example, during breastfeeding, while cuddling, while petting an animal. And so I envisioned the work as an activist intervention to spread affection, physical closeness, attachment, to combat alienation, disconnection, and hate. I imagined a narrative world neighboring our own in which the virus was developed and gained mass adoption. And I'll share a bit of that story with you today. A biohacker becomes disenchanted with the way things are politically and personally and endeavors to change the world. She engineers a love virus in an effort to combat hate and alienation, to make herself feel more connected. In addition to increasing a person's oxytocin levels, inserts a gene for protein as a reporter, which leaves a telltale pink-red glow in the skin of the infected. The virus works, and a biohacker can see the invisible production of oxytocin through its genetic link to the glow of red fluorescence. She loses interest in her phone and begins to feel better, more social, more affectionate, cuddly. She finds herself suddenly holding hands with strangers. Her friends catch on and they want in. The biohacker and her friends begin an underground cottage industry producing the lovesick virus. They encapsulate the virus in glass tubes shaped like the oxytocin molecule that are also charms. The virus is not to be taken lightly. After all, it changes your DNA forever. It's irreversible. New rituals emerge, breaking open the tube like a cyanide capsule, pouring the content into one's mouth, holding it for a minute, incubating, and then swallowing. This is followed by a ritual and polyphonic group chanting of the oxytocin proteins, CYIQN, CPLG. It begins as a cult phenomenon, but catches on with a speed and reckless audacity that you only see in people facing the end of the world. Glowing red symbolizes that you're part of the new growing movement of connection. News feeds feature models and actors splayed on the grass, entwined in a loving embrace with the subtle red glow of one who has been infected. A new group dance style emerges, the tangle. Bodies enmeshed hand in hand, arm in arm, leg in leg, shapes subtly shifting in place. All clubs now feature electric blue lights to enhance their glow. And family changes. Prejudice based on blood gives way to radical openness. The people who are with you at any given moment become your family. And there's a certain return of rituals from the past, holding hands while walking down the street, sometimes in huge groups, before meals, while dancing. And then there's the rejection of devices, giant piles of phones, tablets, laptops, flat screens, and a few VR goggles fill city streets like barricades in the French Revolution. It becomes the thing to demonstrate how over digital you are. There is, of course, the resistance, and they retreat even further into their parallel mediated world. But most people actually leave it behind and come together. We called it becoming one. So I worked on this piece, Lovesick, before COVID. Clearly, I think no one would be allowed to work on that project now, given all that has occurred. But it is ironically at the same time, perhaps exactly the thing that we're going to need coming out the other end. It remains to be seen. So this is the question that I ask, how after a virus has ripped us apart. I think Lovesick anticipated. It was already drawing on the feeling of this kind of digital alienation that, of course, preceded COVID, but has been so much amplified now that we can't even have these kinds of events like we're having today together, that so many things have turned even more remote, even more virtual. And so I really wonder how that will go on. And I'd love to talk also more about it and maybe hear your thoughts on how you think that that will evolve. But Lovesick is this gesture for invoking, wishing for a future that's different, a future where people do come back together physically and have a kind of intimate connection that is not mediated by digital technology. So the last work that I want to share with you in this presentation is Spirit Molecule. And this work poses the idea of a genetic memorial. This is a collaboration with the artist and psychoactive botanist, Philip Andrew Lewis. The project consists of a series of experiments that imagine a future of biotechnological mourning as we engineer a lost loved one's DNA into a psychoactive plant and imagine that this is consumed in a final journey of intimacy with the other. Spirit Molecule has taken place in many different incarnations, so there's been already 3 different exhibitions of this work, each of which is different, and there's a 4th version of it that we're working on now. But each of them turns the gallery into a living laboratory where legal psychoactive plants drawn from the region of the installation, things like morning glory, passion flower, tobacco, liverwort, become subjects of experimentation in genetic manipulation. The genetic material which is chosen for implantation is a 400 base pair bit of mitochondrial DNA, which is passed down matrilineally from great-great-grandmother to great-grandchildren, grandchildren, etc. And it's the same DNA that's often used for tracing maternal ancestry. The film which accompanies this laboratory installation tells the story of one such person who wished to transform their lost loved one, Ginny, into a psychoactive plant to connect with her one final time. And I'll share a bit of the beginning of the film with you. So in the interest of time, I'm just going to pause it there. And I'll say Spirit Molecule is a narrative of love and loss and thinking about bioengineering as a process of grieving. The work invites the visitor to contemplate whether they would like to be transformed into a plant, or perhaps if they would immortalize a loved one in such a way. And generally evoke this thinking about how these kinds of relations, this is what all of these works do, thinking about how our most personal connections to each other, our intimate relationships are changing and will continue to change as biotechnology becomes an increasing part of our everyday lives. And this is just a final frame from our most recent experiments. These tobacco plants, which are an early stage of experimentation, this is called Spirit Molecule Dorothy, and these tobacco plants contain the mitochondrial DNA of Philip's grandmother, Dorothy, who was alive at the beginning of the project but has since passed away. So the project has taken on a very intense and personal significance for us as well. So I'll leave the slides at that, and I want to share just one last bit with you. This is a clip. This is a kind of teaser for a new project that I'm working on called Xeno in Vivo. It's a meditation on interspecies relations and a look at the field of xenotransplantation. And I'll just share this with you directly.
Tucked away in the forest past the fields outside Munich, a team of scientists is designing a new future pig, a pig with a human heart. It isn't really a human heart, but it isn't a pig heart either, at least not as pig hearts were before. These hearts are grown to be transplanted into human bodies whose own hearts no longer function. Are you still other if you become closer and closer to me?
Thank you.
Thank you so much, Heather. You have given me, I've been thinking about this work in preparation a lot already, but seeing the work and hearing you speak about it, I just have a lot of thoughts and questions. So my first question was, we were just talking before, it was going to be my last thought or question, but I want to start with, do you, because it's such a big topic, it probably could just feed us for the next 15 minutes. But anyway, do you see your work as dystopian or utopian? And I know in previous interviews you have responded like, oh, it's up to the viewer. And so that's one thing. And the other is, I was slightly, like my brain was slightly confused if this was a real project or is it like fictional? Is it sort of in between? You know, are you the protagonist? So those are
Or
a few
the
things
antagonist. Yes. Yes, I think that
No,
these are real tensions that are in the work. So the utopian, dystopian.
Yeah, and just one more thing, which you just say that it's for you, it seems like part of the motivation of doing these projects, which are all like amazing, is that is kind of expressing your own conflict with all of it. Right? So,
Yeah.
Anyway.
Yeah, that's exactly it. That's exactly it. And so the point with utopian, dystopian is that it is, of course, both. I mean, almost every technology that I work with contains the possibilities to go in either direction. And these things are really complex, and there's no easy way to say that this thing is good and this thing is bad, or that this thing should be, you know, I don't think that there's any technology I've encountered in my work, which isn't to say that exists in history or in the world, but in the work that I do, that I would say is just bad and should be banned or something like this. I mean, it's always more complicated than that. And so what I hope is that you see through the work that there's kind of both of these things lurking there. So take lovesick as an example. So on the one hand, it has a strong utopian element. I mean, it is this device for thinking about what would it mean to reconnect? What would that look like to move away from a digitally mediated society and think about something else? What would it mean to use biotechnology to make ourselves more loving, more caring, affectionate to each other, rather than more distant, as technology has mostly done so far? But on the other hand, there is, of course, the fact that I made this virus in a lab in a few months. That is kind of a crazy thing. I mean, that you can make a virus that will change human DNA forever, basically, in a few months. And well, I put in the oxytocin gene, which is probably not going to hurt anyone. I mean, that was a choice. That was an artistic choice. Obviously, anyone could put in whatever gene they want and have that be inserted instead. You could put in rather a gene to make people less empathetic. And so it always also hints at these more dangerous, dark possibilities that are also there, that are always there next to, alongside, and really maybe even infusing the hopeful things as well.
Right. So that just made me think about, I think you're also alerting all of us to the, just reiterating what you said, alerting us to the possibilities of science and the positive things, and the, the dark side, which is genetically modified, name your product, including, people. So, I think that's seems to be very much what you're thinking about. And also, I think I was going to ask you, how has your work changed since COVID and so on? But, just hearing that lovesick virus and, was developed before COVID, and now we're developing, the scientists of us are developing as quickly as they can, a vaccine to viruses. It just seems has anyone, I'm just curious if any COVID researchers, do you know anybody, or have anyone connected with you? Curious.
Yeah, I know. So a few of the scientists that I teach with at school definitely are working on COVID related projects. And so I have a good relationship with a scientist, Dewey Hagborg. He teaches at NYU Abu Dhabi, and we've worked together. He's actually helped me a lot with getting access to different labs and different kinds of genetic analyses and things like this at school. And he developed a process for doing saliva COVID tests, for example. And so I know several people who are working on COVID related things. Not that I have necessarily intersected that directly with my practice in any kind of material way. Although certainly conceptually, thinking about where the world is going, and I have great concerns about the creation of borders, and I have definite concerns about the creation of things like vaccine passports and what that means for us biopolitically.
Yeah. It's all, I don't think your work provides answers, but it's very rich with questions, I think. Just one thing I want to mention, which we were talking about earlier, which is I'm fascinated with how narrative, and pictorial your work is, and your writing. So it seems, the Lovesick project is ripe for a movie, which I think, and in a way, you're kind of you are the protagonist in your work, and I find it, it would make a great story if you kind of kept going.
I would love to do that. If there's any, producers, filmmakers in the audience who would like to work on that with me, I would be really happy to take the Lovesick story and kind of scale that up. I think I agree. It is something that I think a lot of people could connect with in this moment in particular.
Right. And, I was just thinking also, we're a limited group of people, and if a film or some kind of documentary was actually made, that would be a way of really, for the layperson, help them understand science, basically. The other thing, I just a response to the work. I'm sorry, I don't remember what it's called, with the plants and the DNA.
Spirit molecule.
What's that project?
Spirit molecule.
So I've been rereading Botany of Desire by Michael Pollan. I don't know if you're familiar with that book. And I think he would really like this project, because he has a chapter devoted to marijuana, and he talks about all kinds of things and how our relationship to plants and so on. I just think it's very interesting. He talks about what you're doing with your plants and ancestors, and all that.
I should reach out to him. Yeah, that's a nice suggestion. I hadn't made that connection myself, but that's a good one.
Let's see.
I can jump back to the unanswered half of your first question, which was about how real the things are. So I didn't address that yet. And so the question of real, it is, however, like the question of utopia, dystopia, which is that it is real and it is imagined at the same time, of course. So everything is based in real science. It's based in real hands-on things that I do in the lab. So I work with scientists, I go into the lab, I do experiments, I research the things, I understand what's happening, I understand maybe what's happening next, what hasn't quite happened, and then try to anticipate that, what those next kinds of steps might look like, and reflect that back through my own personal life, things that I'm thinking about, dealing with, feeling, and then try to kind of filter the science through that to create something that has a personal, evocative, emotional connection for an audience.
So therefore, the data kind of through that lens becomes subjective, is subjective, as opposed to if Bang Wang was here with us this morning, he, all his data analysis has to do with, supporting the Broad scientists who are doing research on, whatever, COVID, for example, so he has a very specific, the data has a very specific role, as it does with most scientists, although they might have feelings about it, they are subjective since they're people, and they're dealing with, as best they can, making that data objective. And you kind of just jump over, and I just think it's really pretty amazing the work you've done. So, are doing. I wanted to ask you about. Hold on a second. I'm sorry, I'm looking at my notes. I'm sorry, the Probably Chelsea project. Can you give us the backstory of how that even came about? Because it's pretty interesting.
Absolutely. It's a long story, which is why I didn't include it in the slides in detail. But so basically, so years back, so that was in 2014. In 2014, so I was contacted by Paper Magazine. They were doing an interview with Chelsea Manning. So Chelsea had already been incarcerated for years in a federal military prison. And at the time of her sentencing, she announced her gender transition. So she was arrested as Private Bradley Manning, announced her gender transition as Chelsea Manning. And the other background in that, of course, is that she was imprisoned as a whistleblower. She was imprisoned for making public information that showed the prevalence of torture and civilian deaths in Afghanistan and Iraq wars. And so she was incarcerated at her sentencing, announced her gender transition. And at that moment, her image was censored from the public. So no one was, after that moment, allowed to see her, allowed to visit her. And so she was effectively had her image suppressed. And so Paper Magazine was working on an interview with her through the mail, and they wanted some kind of picture to accompany that, but no one could visit her, no one could see her. And no one had seen her. In a way, no one had ever seen Chelsea Manning, because her gender transition was after the point at which her image was suppressed. And so they came up with this idea of doing a DNA portrait. They'd seen my work, they talked to Chelsea about it. Chelsea had also seen my work, and she was excited about the idea too. And so ultimately, Chelsea sent me in the mail cheek swabs and hair clippings, and I walked through the same process that I went through with Stranger Visions, except that we had had this conversation about what to do about the genetic sex parameter. And so for me, this was a perfect opportunity to complicate the genetic determinism that's inherent in this kind of genetic portraiture, and to kind of use that as a way of opening up these possibilities, this possibility space of DNA data interpretation. And so first, we created two portraits to represent her that we had side by side, and one was algorithmically neutral, and the other is parameterized female. And these sat side by side on the wall, and we're hoping to bring people into thinking about this kind of reductionism around gender stereotypes and the face and DNA phenotyping and these things. And then, long story short, so we created this comic book also while she was in prison to advocate for Obama to commute her sentence. And the comic book ended with this speculative frame. So it was a kind of comic book that talked about our collaboration. And then it ends with this last scene where Obama commutes Chelsea's sentence and she's freed, and she comes to the gallery and sees her portraits in person for the first time. We published the comic book, and that very day, Obama actually commutes her sentence. So it's like this magical incantation, really. It's totally amazing. And then, so probably Chelsea, the work that I showed you a quick video clip from, so that was this kind of celebration of her release from prison. So we were offered this exhibition to celebrate her release, to show the portraits finally, to allow her to have this experience of really coming and seeing her portraits in person. And so we took that as an opportunity to scale that up and to show not just two, but these 30 different possible Chelseas that could come from her DNA.
Thank you for sharing that, because it's totally, it's totally amazing. This is a good moment to invite the audience. Actually, Eduardo was going to read some questions and thoughts from the chat to you.
Yes, awesome. So as I start looking through the questions, we're gonna start allowing everyone's video to be allowed to be turned on. I would like to ask you all to please make sure that you type your question in the Q&A. And if you do want to ask your question directly to Heather, you can just raise your hand and we'll unmute you. So we're gonna bring everyone. Just bear with me here.
Nice to see you all. Hello, everybody.
If you do not want to turn on your camera, that's totally fine as well. We just want to see all your faces before we exit out. Okay, awesome. So Heather, our first question is from Emily Garfield. How do you think of your work, if at all, in the lineage of science fiction, which is also historically about integrating the possibilities of science?
Yeah, that's a good question. So for me, I think in terms of my own journey, I came less from science fiction and more from speculative design in terms of that side of things. So I was very inspired by the work that I saw coming out of Tony Dunn and Fiona Raby's lab at the Royal College of Art, 10 years ago or so, 10 to 20 years ago, that they wrote about in their book Speculative Everything. So there was this kind of moment where there was so much of this speculative art and design making happening that was very engaged with thinking about the futures of science and specifically the futures of biology. That really came out of their lab. And so I was really inspired by that, and then also by bioart, which is not on the speculative sci-fi part necessarily. It's more on the kind of critical side. But for me, kind of fusing those two things together was what I found exciting. And so in a way, I've kind of come to science fiction the other way around. So not that I grew up reading a lot of it or anything like that, but that actually now I find it very interesting to read and to watch films and things like that. But it sort of came through the other way.
Okay, we have another question from Sarah Putnam. She says, amazing work, such essential questions. Where, how can we see more? I would love to see the whole entire films.
Oh, yes, absolutely. So for the most part, the films, they do cycle through galleries and things like this. So I haven't figured out a good distribution platform yet. I would love to actually have them, available for streaming and things like this, but I haven't figured that out yet. So the best way is basically to contact me And I can just put my studio email in here. And, I would love, if anyone wants to set up like a screening event or something like that, invite friends. I would love to see the thing circulate more.
Okay, we have a question from Ellen. I'm going to go ahead and ask Ellen to unmute herself, please.
Thank you very much. This was really fascinating, and I have to admit that there were moments when I was overwhelmed with fear, truly. And so many things were racing through my mind. I've been reading speculative fiction for years, and several years ago, I read Margaret Atwood's book about speculative fiction and devoured it, really. And, her books also frighten me and attract me at the same time. So it's like you know what I'm saying? Anyway, but the piece about oxytocin is especially compelling because it really brings into question the reality, authenticity, whatever, of our human emotions. Like when we're in love, are we really in love, or is it just a hormone? And I bet everybody here would be thinking the same thing. And so, that's something I've actually thought about for years when I became aware of hormones and emotions, which goes, back decades. And so was that in part what you were striving for in that piece, was how much is it the hormone, what, like the relationship between hormones and emotions, and like, what are we actually in love with? And is it love? Like, what's love got to do with this, kind of thing.
Yeah, I like this interpretation a lot. It's not necessarily that I was explicitly thinking about exactly that scenario, but more generally that in thinking at the projects, I'm also often thinking of them as metaphors and thinking about my own relationships and what do these things mean, and how do I imagine technology playing into that, or standing as a metaphorical layer. And so I think that's a very interesting interpretation, and I like that a lot. But I would say for me, the primary thing that I was thinking about is more this biotechnological future aspect. So thinking, how will we change ourselves? Would we change more loving to be less loving? Would you find it ethical to create a virus to make someone fall in love with you? These kinds of things.
Thank you.
Okay, we have another question from Rebecca Reynolds. Did the Lovesick virus work? Did you actually try it on people?
So there have been no human trials, no animal trials either. So we just worked in vitro only. So we did experiments on cells, and we did do analyses of those. So we did an antibody test to see that the oxytocin was being produced by the cells. But clearly, to go all the way to making an actual drug that you would release would require a lot more work and years of testing, as we're all very familiar with now from COVID vaccine processes, all about these things today. But so there would be a lot more required to do that in a really legitimate way, definitely. But it was kind of the first steps of experimentation and seeing that it worked.
Okay, do we have any other questions? Anyone would like to raise their hand and speak directly to Heather? Okay, that concludes our Q&A.
Yeah. If I can ask another question, has there been a silver lining for you? Maybe that would be my closing question. In any aspect of this year, or your work in Abu Dhabi, the people you're working with, your own feelings, has something kind of, encouraged you to pivot in a way that you might not have?
Yeah. I mean, I would start by saying that it's been a really, really hard year. I know I'm not the only one that feels that way. It's been really devastating on multiple levels. So it's been, actually, I would say for me, it's been the most challenging year of my life, without question. and definitely not the most productive. Definitely not the most artistically productive year or artistically successful. You know, probably it's like the least exhibitions that I've had in years and these kinds of things. So that's been really hard. The one thing, I guess, if you can call it a silver lining, so I did do a body of work closer to the beginning of the pandemic that was about kind of thinking about time, thinking about viral time, and using using HTML as a kind of narrative device for thinking, for exploring this kind of storytelling around the experience of being a virus. What would it be like to be a virus? And I spent some time doing certainly more drawing and painting and things like this than I normally would. So it's, I don't know if for me it's really a silver lining. It was sort of like a coping strategy for just getting through the days. Yeah.
Totally understood. I think many of us figured out some kind of coping mechanism to get through this, you know, continued difficult moment. And but you know, I am heartened that we can, you know, we Catalyst can do programs like this and have people like you, Heather, be with us on a Wednesday morning. It's really terrific and, you know, so thought-provoking. So and thank you for sharing your email in our chat. So and being generous if people want to get in touch with you. So in my own experience, I was just thinking about my Michael Pollan idea is, even though you think someone is like really famous or, you know, unapproachable, usually people like, you know, they're interested in people that are interested in what they're thinking about. So I would just, just say that generally. So we're almost at the end of our, sorry, go ahead.
No, I was just saying, yes, I think you're right. I think that's true. And it's a really an interesting suggestion to reach out to Michael Pollan.
Anyway, we are at the end of our hour, and I want to thank you again, Heather, for sharing with us with your amazing work and thought-provoking work. And I'm really glad to see you again, since the last time was 2013. And just for everyone to know, our next Catalyst program is May 26th with Ani Patel, who is a wonderful neuroscientist who thinks about music and the brain. And he's been working with a cockatoo. I don't know, I've seen it sort of circulate on YouTube. Snowball, I think, is the name of the cockatoo. And what he's thinking about and researching is, are there other species that have a sense of rhythm? And this particular cockatoo does. He can kind of anticipate a beat. And anyway, Ani is pretty great. And he will be on May 26th. So with that, I think we'll end here. And thank you all for coming. Great questions and good thinking. And we'll see you May 26th. Thank you.
Thanks, everyone. Thanks for having me, Deborah.
Thank you.
Bye.
Bye, everybody.
Have a great day.
Thank you. Thank you, Eduardo.
Take care, Deborah. We'll talk later.
Okay.